Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone.
Waalaikumsalam warahmatullahi
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, everyone. And the Honorable, our special guest today, Professor Ahmed Sherif Atia. Thank you for kindly joining us. And greetings to the faculty members and students. Thank you for joining us this afternoon. Yeah. So my name is Farida, and I'm going to be a moderator for this meeting. And our topic today is update on antimicrobial agents, the biomolecular approach, and potential drug targets. I think this will be an interesting topic because antibiotics are widely prescribed by the doctors to treat bacterial infection by killing and or preventing them from multiplying. There are various antibiotics are available, but how is the mechanism of action to destroy or slow down the growth of bacteria, and what happens inside the bacterial cells after antibiotic treatment, and what are recently studied potential target therapy for antimicrobial agent? So to answer that question, today we would have a guest lecture from an amazing expert and researcher, Professor Ahmed Sherif Atia from Cairo University. Yeah. Good afternoon, Prof. Atia, or maybe good morning in Cairo. Yeah.
Good afternoon. Thank you, Dr. Farida. Thank you.
It is so good to have you here. Thank you for joining us today.
My pleasure.
So before Prof. Atia starts the lecture, it is good to know about him and his research expertise. So let me deliver his brief biography. Professor Ahmed Sherif Atia works as professor of microbiology and immunology at the Faculty of Pharmacy, Cairo University, and has an adjunct faculty position at the School of Pharmacy, New Giza University. He obtained his PhD in molecular microbiology from the University of Texas Southwestern Medical, the USA, working on the molecular aspects of microbial resistance to the complement system. Working as a postdoctoral fellow at Vanderbilt University, he identified new microbial therapeutic targets and novel host antimicrobial mechanism using cutting-edge technologies. Professor Atia's current research focuses on identifying novel microbial therapeutic targets, developing new vaccines and biotechnological products, and discovering novel non-traditional antimicrobial agents from natural sources through synthetic chemistry. Prof. Atia's work is highly recognized as he has been awarded several prestigious awards from both local and international entities. So, Professor Atia, would you please start the lecture? The time is yours.
Okay. Thank you very much, Dr. Farida, for this nice introduction and for the invitation. It's my pleasure and honor to be with you all today. And hopefully we'll have like a fruitful discussion about a very important topic that we would like to share with you in this lecture. So I'll start by sharing my slides. So is the slides showing up on your screens now?
Yes. Yeah.
Yes, sir.
Okay, sure. So let me close the video and like it will be like better for the streaming. So So is it now in the full screen mode for you?
Naam, dapat full screen now.
It's not full screen?
Full screen, sir.
Okay, so again, it's my pleasure to be with you all today. We'll talk today about an update on the antimicrobial agents, focusing on what we call it a potential drug target and how we can use biomolecular approaches to identify those new targets and potential one. So this is like an outline of our talk today. We'll start by having a brief introduction on infectious diseases. Then we will move to antimicrobial agent, have a historical background. Then most of the talk will talk about the mechanism of action of already known antimicrobial agents we are using in the clinic. Then we'll talk about a crisis we are having now, which is called the post-antibiotic era. Then we'll explore what are the potential solution for this problem by talking about alternative approaches. We'll talk a little bit about what is already in the pipeline, trying to find new antibiotics. And finally, we'll end by a story, what we call it a homemade drug, like something that we have developed in our lab here in Cairo University. And I think promising and might be interesting to you to know about it. So first of all, we start by an introduction. So what are infectious diseases? Infectious diseases, they are illnesses that are caused by harmful agents. We call them pathogens. They get inside our body, and by the way, they can also be pathogenic for animals. And the most common causes that are categorized as pathogens are either viruses, bacteria, fungi, and parasites. Infectious disease usually can spread from person to person. However, they can be transmitted by food, contaminated water, or even by insects like mosquitoes, which transfer some infectious disease. Some infectious diseases are very minor and they are self-limiting. However, some infectious diseases are very serious and they could be life-threatening. So, if we want to know the impact of infectious disease, here is like on the left of this slide, as you can see here, the biggest 10 threats toward health in 2019. And I have just picked 2019 for a purpose. This was before even the COVID-19 pandemic that crippled the world throughout the previous 3 years. And if we check these 10 threats, we will find 6 out of the 10 things are related to infectious disease. We are talking about influenza, antimicrobial resistance, Ebola and high-threat pathogens, vaccine hesitancy, dengue fever, and HIV. And again, this is back in 2019. Out of 10 biggest threat, 6 were related to infectious disease. How about moving to 2021, like 2 years later, while living throughout the pandemic of COVID-19. As you can see, if we check the greatest risk in 2021, you can see that infectious disease has reached the top of the list, making impact on all over the world. This is like world out thing. And of course, many of us, if not all of us, have suffered some impact of this pandemic we are suffering from 2020 up to very recently. So you cannot defeat your enemies, which are causing those infectious disease, until you know who they are. So who are our enemies? Our enemies can be classified into 2 groups. A group we call them living, or what we call them cellular, and a group which is called acellular or non-living. For example, for the living one, we have the major one, which are bacteria, which are classified as prokaryotes, and they cause many diseases, for example, leprosy, or fungi, which causes athlete food, protozoa, like those causing malaria, and parasites or helminths, like the tapeworm. On the other hand, what will be classified as non-living or acellular pathogenic agent, we have viruses like the COVID-19 virus or the virus causing COVID-19, and also the HIV virus causing AIDS, and also we have infectious proteins like the one causing, we call them prions, like the one causing mad cow disease. So those are the enemies. What do we use to fight them? Who are our soldiers in this fight? Those are we call them antimicrobials, which will be the focus of our talk today. So, what are antimicrobial in a very simple overview? They are compounds that used to combat pathogens. They are classified into 4 major groups. What we call them antibiotics. This is the major one, and will be most of the focus today about it. They are known as antibacterial. They are used to prevent or treat infection caused by bacteria. For example, we have a urinary tract infection, we have tuberculosis, we have sexually transmitted disease like gonorrhea. Those are caused by bacterial pathogens, and they are treated with antibiotics. For the other group, we have antivirals. They are used for viral infection like HIV infection, flu, chickenpox, and of course we got the COVID-19, which is caused by the virus, so we treated by antiviral agents. We have antifungals, which are used to prevent or treat infection caused by fungi, and here we have again the most known one is what's called the sleet food. Antiparasites, we use them for treating and curing infection by parasites like malaria and so on. Antimicrobial drugs, if they are working and the pathogens are killed by them and infections are clear, so we say this antimicrobial agent is working well. However, if the agent, like the bacteria, the fungi, the virus, or the parasite, was mutated or changed itself and become non-susceptible to killing by our agent, this is what we call it antimicrobial resistance. So the pathogens are resistant to killing by the agent we have developed to treat infection and get rid of this pathogenic microorganism. And this is a very hot topic. Maybe we can talk about it in another session. So we'll start by a historical background of antimicrobials and a little bit focus on antibiotics. So usually when we talk about antimicrobials, we think only about the 20th century. However, during the 20th century, we found this discovery. These Nubians, like the Nubia area, Sudan, South Egypt, excuse me, they found that in their bones, when they have done the analysis, there is the antibiotic we are currently using called tetracycline. So, and actually it was present in very high concentration that makes the scientists who make this discovery assume that the people living at this period 2000 years ago were consuming a lot of amount of this tetracycline. Even they considered it was using like a beer or a beverage that you are taking or consuming large amount. So they are very old. People know about antibiotics very old. However, in the recent history, if you are talking about only the 20th century, we got Joseph Lister, who is developed what we call it antiseptic revolution. He found that this molecule, maybe most of us are already expert in chemistry, studying in pharmacy school. This is phenol, or what we call it carbolic acid. Joseph Lister found that using carbolic acid or phenol can kill microbes that could be introduced to patients during surgery. So it was very good at killing microorganisms contaminating the surface of a surgical operation theater. So it was very very effective in decreasing the complication of what we called surgical wound infection. However, carbolic acid is very harsh. If you end up like having carbolic acid in your internally, like this is an esophagus or duodenum or stomach, who for somebody who got exposed to carbolic acid, as you can see, there is a severe damage. So giving carbolic acid to treat infection that's already present in a living organism, especially human being, was very, very dangerous. Actually, it could kill this patient. Accordingly, the journey to find something that can kill microorganisms inside the host without harming him started. And here we started by early 1909, at the beginning of the 20th century, we got what we call it the magic bullet. The magic bullet was a terminology that is used a lot of time from now on. However, it was first introduced to mean targeted drug, something that can be used to kill harmful microorganisms without causing harm to the host. And who developed this was Paul Ehrlich in 1909. The drug was called Staquorsin, and it was very effective as the first effective antimicrobial drug in the Western medicine. It was used to treat syphilis, with a sexually transmitted disease. However, it was not 100% effective, and the treatment required a very lengthy period of time, even up to 2 years. However, Nobel Prize was awarded to Ehrlich, and also he was portrayed on the 200 mark Deutsche Mark back then as an honor for him, because it was a breakthrough for treatment. So even Hollywood make a famous movie about magic bullet of Ehrlich, and it was very interesting, indicating how this a lot about what we about treating bacterial. And then the miracle drugs. They are sulfa drugs. They are discovered by a German scientist, Domag, and it was used to treat many infections. And actually, it caused a revolution in the medical field, as it saved millions of and thousands of lives, especially during World War II, which started 4 years later after the discovery of this drug. Finally, this is the most famous antimicrobial agent we know about. It's penicillin. Penicillin is usually associated with the first scientist here, while 3 of them got the Nobel Prize for the development and the discovery of penicillin. However, Fleming is taking most of the credit. He is the one who discovered penicillin accidentally. We'll see in the next slide. However, without the effort of Florey and Chain, we will not able to find penicillin in the clinics. So they are the one who work in making penicillin, which is actually the first antibiotic to be known and very well studied. However, without the efforts of Florey and Chain, there will never be a drug that we can use. So what happened? Like Fleming was working with a bacteria, a pathogen called Staphylococcus aureus, and he discovered that he one day left his lab early, and he was not very tidy in doing his experiment. So one of the lids where we are growing the bacteria was a little bit open. So this introduced what we call it a fungi. A fungal spore entered the plate, then it grew. When it grew, he discovered, he noticed that the area surrounding this fungi is not containing any bacteria, although the rest of the plate contains. So what caused the killing of the microorganisms, staphylococci, in this area turned out to be penicillin, which is like a lifesaver during World War II, and up till now we are still treating infection. Maybe almost 100 years later, we are using penicillin for treating infection. So what are antibiotics? So they are medicines that we are using to fight bacterial infection. They either kill the microorganism, we call those bactericidal antibiotics, or we are trying to make their growth very hard. They are inhibiting the growth of the microorganism, and those we are calling them bacteriostatic antibiotics. So we either kill or inhibit the growth. There are different ways to classify antibiotics. One of them is just according to the origin, where we obtained this antibiotic or this compound. According to this method of classification, we have only 3 sources. Either a natural source where a fungi or another microorganism is producing the compound, as the case of penicillin. In the case of Alexander Fleming, the fungi was producing penicillin, and the penicillin was the antibiotic that was killing the bacteria. However, due to this natural source and the presence of this compound already in the nature, so usually this is associated with rapid resistant development, because bacteria in the environment is already exposed to this antibiotic or this compound in the nature with large amount. And also regarding toxicity to the host, they are the most toxic one. As a famous example here is penicillins. Also, we have the streptomycins and tetracycline. So if we took this natural source-driven antibiotic and perform on it a biochemical reaction in the lab, we develop what we call them semi-synthetic antibiotics. So the origin is a microorganism. However, we do a biochemical reaction on this product and produce a new antibiotic. This new antibiotic have lower toxicity and more effective in killing microorganisms. So this is a very good development. So many of the penicillins derivatives, so those are derivatives of penicillin, which is a natural one, and also cephalosporins are categorized under this group. We call it semi-synthetic antibiotics. So the third class is artificial, or what we call it fully synthetic antibiotics. This is a compound that is purely produced in the lab. So we are doing chemical reaction to produce compound like what we have here, the quinolones. They are highly effective. They are very low toxicity to the host compared to the other antibiotic, and guess what, they are the most expensive because this reaction will cost a lot of money to produce a molecule like this. So this is the way or the sources of antibiotics, either natural, semi-synthetic, or totally synthetic or artificial drugs. So in order to follow out the evolution of antibiotics, we have this timeline. We started by the 1909 discovery of sulfurcin, then we have the sulfonamides, then we started to have a lot of antimicrobial agents. And as you can see here for the key of this slide, those which are written in green, they are from actinomycetes, which is a type of bacteria. Those which are written on blue, they are driven from other types of bacteria other than actinomycetes. Those in purple are fungal, those in orange are synthetic one. As you can see, as we move on this timeline, the period from the mid-40s to the mid-60s, we got a lot of discoveries. Very large number of antibiotics were discovered or developed. So they called this period the golden age of antibiotics. Moving away from the golden age and getting from in the 70s, 80s, as you can see, the number is going down. So till we reach what we call it the discovery void, that we were not able for almost 40 years to find new classes of antibiotics. We are either getting the old one with small modification or something that's working the same. Not a new class for almost 40 years. Part of this, actually, it's like what we call it financial incentive. Pharmaceutical industry is not interested in finding new antibiotics. Why? Think about it. If you are having urinary tract infection or respiratory tract infection, and you go to the doctor and he prescribes for you an antibiotic, how long you will take this antibiotic? A week, 10 days. However, if you are diabetic, if you are having hypertension or a cardiac problem, you will take drugs forever. So the incentive is very low. Moreover, the problem is complicated with antibiotics because we are facing the problem of what we call it antibiotic resistance. So few years after the introduction of the antibiotic in the market, bacteria is resistant to it. So people stop taking this drug and it is trash. So the financial incentive is not making the pharmaceutical industry very interested in doing research and development of new antibiotics. And this is a big problem. We suffer from it for a very long period of time. So how do antibiotics work? So in order to understand how we can find new drugs, how we can develop potential drugs, we need to understand how the ones that already we have is working, what is the mechanism of action in order to be able to understand how we can develop this and evolve it in a more better solution. In general, we have 5 types of potential mechanisms. The first one is dealing with inhibition of cell wall synthesis, and here we have 5. So what we have here on the right, those are the monomers building the cell wall of the bacteria. The cell wall of the bacteria is a very important component for the bacterial cell survival. If the bacteria lost its bacterial cell wall, most probably it will die due to lysis. So this is a very essential component for the bacteria. So what we have here, we have an enzyme that will join these 2 monomers. This enzyme is called transpeptidase, or what we call it penicillin binding proteins. Penicillin binding proteins is the target of penicillin and cephalosporin. So if we have penicillin or cephalosporin, they will target the transpeptidase enzyme and will prevent this reaction from getting on. So the bacteria will fail to form its proper cell wall and it will die. Another group of antibiotics called glycopeptides, the most famous member in this group is vancomycin, is also targeting another step in the building of the cell wall of the bacteria called transglycosylase, which is again important for the polymerization of the components of the cell wall of the bacteria. Preventing transglycosylases again makes the formation of the cell wall improper and the bacteria will be killed. So this is the first mechanism, inhibition of cell wall synthesis, which is very effective, and many of the antibiotics we are using or used to use are targeting this step. The second possible mechanism is what we call it blocking of protein synthesis. If you recall a little bit of biology, the ribosomes are the protein factories of the cell. So ribosomes consist of a small subunit and a large subunit. The small subunit in the bacteria, we call it 30S, while the large subunit, we call it the 50S. For example, here we have antibiotics which are called aminoglycosides. When they bind to the 30S subunit, they can block the initiation of protein synthesis, so the protein synthesis is totally not working, or they are blocking further translation and elicit premature termination. So the synthesis start, however, it will never end in the proper way. Finally, it can be a faulty protein synthesis, incorporation of incorrect or wrong amino acid. So we end up with a protein that is like not working right. So here we have another example, also binding to the 30S. To the 30S ribosomal subunit, and we have here the tetracycline. The tetracycline binds to the 30S subunit and blocks the transfer RNA. Here is the transfer RNA bringing the Here is the transfer RNA bringing the new amino acid to the ribosome to be incorporated in what we call it the A site in order to join the protein synthesis process. Tetracycline prevent this step, resulting in protein synthesis inhibition. We have also other antibiotics which are binding to the 50S ribosomal subunit. For example, the chloramphenicol bind here to what we call it the peptidal transferase site, or the P site, and this will prevent what we call it transpeptidation, the joining of the new amino acid coming in the A site to the nascent polypeptide chain. Finally, in this group, we have the macrolides, or something like the erythromycin, where the erythromycin or the macrolide inhibit what we call the translocation step. Here, the new amino acid has been already joined the peptide that is forming, and it needs to move to the B side or the donor side. However, the erythromycin will block the movement, will prove the translocation of the transfer RNA and the new peptide to the other side, again blocking protein synthesis. And as you can see, if the bacteria is not able to synthesize its protein, this is a lethal effect. This will cause the bacteria to be killed or not able to grow. Another approach or another mechanism for nucleic acid for antimicrobial is binding to nucleic acids, and we have two targets here: either DNA, or what we call deoxyribonucleic acid. For example, the quinolones, they are targeting what we call them topoisomerase enzyme that are important for the DNA synthesis process, and we end up by malfunctional DNA synthesis resulting in cell death. Or we are targeting RNA synthesis, ribonucleic acid. Here we have the enzyme responsible for RNA production, which is called DNA-dependent RNA polymerase. The RNA polymerase takes the DNA template, which is here depicted in green and red and blue, and use it to synthesize the messenger RNA molecule, which is green. Rifampin, which is a drug used for treating infection like caused by tuberculosis, will bind to the beta subunit of the RNA polymerase, blocking the messenger RNA elongation, and we prevent what we call transcription or RNA synthesis, and again this results in a lethal effect on the bacterial cell. The fourth mechanism for antimicrobial agent is injury for the plasma membrane, or what we call the cytoplasmic membrane, which is very crucial for the living of the cell. If there is an injury in the plasma membrane of the bacteria or even any cell, excuse me, they are lethal. They die. The cell will lyse, and all the components inside the cell will leak to outside, and the cell will die. Polymyxins, which are the group in this class, they are positively charged. They bind to the negatively charged component in the cell wall, which is the LPS, and disrupt the integrity of the membrane in what we call them gram-negative bacteria, and those are very effective against gram-negative bacteria. The fifth mechanism is what we call it anti-metabolites. What is anti-metabolites? We have a metabolic pathway. What is the metabolic pathway? We have a substrate. As a substrate, we have an enzyme that will work on this substrate to produce a product. Now, this product is the substrate for another enzyme that will work on it to produce a second product. The second product now is a substrate for a third enzyme to produce a product, and so on. What we have here is the pathway for the synthesis of purines and pyrimidine nucleotides. Those are the building blocks of nucleic acid, either DNA or RNA. The first substrate is called PAPA, or what we call it para-aminobenzoic acid. This is the substrate that fit in this enzyme, which is the dihydrobutyriate synthase. Here we have the PAPA, the normal substrate, binding to the enzyme, so this reaction will take place normally, and the bacteria will synthesize its DNA and RNA. However, what we have as an anti-metabolites, the sulfonamides, the sulfa drug, the magic bullet that we talked about earlier today, is, as you can see here, it's similarly mimic the para-aminobenzoic acid, but instead of having this carboxylic group, it has this group. So this compound, the drug, will bind to the enzyme, and this will not result in the production of the product of this enzymatic reaction, so it will block the metabolic pathway, and this is why we call it anti-metabolites, which are the magic bullet that was first discovered as sulfa drugs. Okay, so what is common among these mechanisms? We talked about protein synthesis inhibition, nucleic acid synthesis inhibition, inhibition of cell wall synthesis, disruption of cell membrane, or blocking the metabolism of the bacteria. What is common among them? That they are targeting essential processes inside the cell. Yet they are unique. What is meant by unique? All these targets are present in prokaryotic cells, in the bacterial cells, and absent or different from counterparts in the human cell. This is what makes them effective in killing them. So is this a problem? This is something good. This is how we spend 100 years using antibiotics, which is targeting all these antibiotic target, either cell wall, protein senses, nucleic acid senses, membrane, or metabolism. However, is this going to work forever? Unfortunately, no, because since we are targeting a very essential process in the life cycle of the pathogen, this will exert a very high, what we call it, selective pressure on this bacteria. So this bacteria is under a huge selective pressure that will lead to the development of what we call it resistance. So let us see this situation. Here we have normal bacteria, which is depicted in blue, and we have only one resistant cell, and this developed just by random mutation in the target of this bacteria. What happens when we use antibiotics? When we use antibiotics, especially we are using them in very large amounts and also using them in non-indication purposes, what happens? The bacteria, which is sensitive, which was the majority, will start to die, and we'll have only a minute amount of the sensitive bacteria, and we still have the resistant one did not die. If we continue to use more antibiotics, what will happen here? We will kill almost all the sensitive bacteria, and the resistant bacteria are starting to get dominance till we end up with totally resistant population. And this is the problem we are facing now. Bacteria is turning into what we call them superbugs. What's superbugs? They are resistant to almost all types of antibiotics that we have. We have a patient and the doctor prescribed for him an antibiotic, and he takes antibiotics and will never get cured. Why? Because the bacteria is resistant, and they are resistant to almost all types of bacteria, all types of antibiotics that we have already in the market. That's why the terminology post-antibiotic era. We are in a period of time that the antibiotics, those magical drugs, those magic bullets, those miracle drugs are not working anymore. So we are facing a very big crisis in the medical field. And as we said, like, we might be talking about this in more details in another occasion. So what are the alternatives? So are we going to sit as we are and we are losing this battle against our enemies, which are the bacterial pathogen? We should be looking for alternatives. And here we move to what we call it the biomolecular approach. For this, we have several avenues. The first one, or the first strategies, is called potentiators. What is potentiators? It's actually a combination therapy. A combination therapy, when we are trying to treat a bacterial infection, instead of using just one antibiotic, we use a combination of two compounds. And we have three possible cases here. We have what we call a Congress combination. We have molecule A and we have molecule B. Both of them are targeting essential process in the bacteria. So what happens now when we take them concurrently, we take them together, we take them as a combination, both of them are targeting essential targets. So this will give you a better effect, and it is less likely to develop resistance to both of them together. So this will allow a better chance for the success of treatment. The other case, we have what we call it synthetic combination. Synthetic combination, we have our normal drug, for example, penicillin or ampicillin, that is targeting an essential target in the microorganism. We have another compound, a compound B, which actually has no target in the bacterial cell, regarding its essentiality. However, it's targeting what we call it a resistant element. For example, an enzyme that the bacteria will produce to break down compound A. So if we combine with compound A another compound that will prevent the bacterial enzymes from degrading A, so we will save A and A will be able to kill the microorganism. So this is what we call synthetic combination. We get synergism, like a compound that's effective and a compound that is protecting the effective molecule to keep its function. The last potential combination here is what we call it holistic approach, and this is very interesting approach. Here we have compound A and we have compound B. Both A and B are not antibiotics. They are not having any target, either essential targets inside the bacterial cell. However, their combination inside the bacterial cell develop what we call it synthetically lethal gene function. When they are combined, although both of them are non-essential targets, they are targeting non-essential target. When they are taken in combination, this combination result essentiality and lethality to the bacterial cell, and this will result in a better outcome for the treatment. So this is the first approach. We are trying to extend the life of already present antibiotic, either by combining them together or combining them with compounds that protect them from harmful effect by the bacteria, or actually finding something that will develop into a essential or a lethal thing inside the bacteria when it's combined with another compound. A second approach for finding alternatives for antimicrobial agents or antibiotic is doing what we call it drug repurposing. Like we have a drug, for example, used for hypertension, for diabetes. How about it turn out to be antimicrobial or antibacterial? So this is what we have as anti-repurposing drug. So here we have an existing antibiotic that's effective against the bacteria, and we know its target. Knowing its target, and this is why it was important to understand the mechanism of action of traditional antibiotics, so we can use this information with the knowledge and doing what we call in silico drug repurposing. We have massive databases of all the drugs that are already approved and they are already in the market and used for different indications, diabetes, hypertension, antibiotics, and so on. We explore this existing antibiotics and try to find if any of them will fit with this target. So if it fits in this target, it could be potentially a new antibiotic, and it could be used as a new treatment, as a new indication for this already drug that's already in the market. This is very advantageous because this drug is already used. So it is have passed all the steps and also have passed all the regulatory affairs that's required for its approval. So it's already drug in the market. So this will like save a lot of time for the development of new antibiotic. And for sure, we have heard about this in the very nearby case. I'm not sure about Indonesia, but I think it was all over the world that during the COVID-19 outbreak, we have done a lot of drug repurposing, and maybe the most famous one was the chloroquine and hydroxychloroquine, which were used initially for treating malaria. They used them for COVID-19, although malaria is a protozoa and COVID-19 was caused by a virus. However, we were repurposing that drug. And we have also other examples for this case. And as you can see, the repurposing could be done in silico. It could be used in what we call network or neural networks, or also it should be tested in vitro, like in experiments in animals, and also it should be tried on clinical trials when they proved efficacy in all these cases. So this is the second approach, trying to, instead, in the first one, we are trying to extend the life of already present antibiotics. In the repurposing, we are trying to extend the spectrum of already existing non-antibiotics for treatment of bacterial infection. How about the third approach is trying to tweak with the immunity of the host using immunological tools, either antibodies or vaccines. They are given to the patient infected with a pathogen so that those antibodies, which are pathogen specific, will enhance the process of phagocytosis. Neutrophils and macrophages will kill the bacteria better when they are covered, or what we call them, opsonized with pathogen specific antibodies, and this is a very effective one. A fourth one is what we call it immunomodulation. Here we are not just using antibodies or vaccines, we are trying to manipulate the immune response within the host. And the example here we have it is for urinary tract infection, UTI. This experiment was done in the mice, and those are the kidneys of the mice. As you can see here, when we are knocking out or using chemicals that are blocking a key immunomodulators inside our body, again, we are getting a protection, or we're getting cure for the infection. And again, in the case of COVID-19, this was a very well used example, which is the immunomodulation. The reason for this is what? Because most of the pathological findings we are suffering during infectious diseases are due to the immune response of our body to the infectious agent, either the virus or the bacteria or the fungi. So if we are trying to reduce the damage, we can modulate the immune response. For example, here's the hydroxychloroquine and azithromycin. Azithromycin is an antibiotic. However, it was used for treatment of a viral infection. What we are using here, we are using its immunomodulatory activity. A terminology that we heard a lot about during the COVID-19 was the cytokine storm. The cytokine storm, we can control it with different drugs, including statins. Statins are those drugs which are used for lowering the lipids in our blood. The triglycerides in our drug, we are using statins for them. So again, this is a holistic approach, trying to modulate the immune response in order to better control the infection caused by another infectious agent. The fourth approach for as an alternative is what we call it phage therapy. For those of you who do not know about phages, phages are viruses which infect bacterial cells. So actually, we can treat bacterial infection by injecting the patient with a phage, which is a virus that will kill the bacterial cells. This is the classical mode of virus. We can use phages and package them with some compounds that will prevent the formation of what we call it biofilms. Biofilms are aggregation of the bacterial cells inside the host tissue. So if we have a compound inside the phage, and this phage was introduced into the infection site, it can help dissolving the biofilm and preventing the infection from getting worse. And also, guess what? We can package the phage with immunomodulatory components, again, to modulate the immune response in the site of infection and causing curing of the infection or less damaging effect. Also, we have another approach called microbiota modulating. What's microbiota? Microbiota is microorganisms which are living normally inside our body, and they are very useful. So if we are suffering from an infection, we can introduce healthy microbiota inside the host. They can exclude the pathogen, giving them no space, no nutrition to survive. They can regulate the immune response so that we can get rid better of the pathogen, or even they can enhance the protection inside our body. For example, we have in the intestinal cells what we have them goblet cell. Goblet cell produce mucus secretion. So the normal microbiota will induce mucus secretion and help protect our intestinal mucosa. Finally, last but not least is what is called anti-virulence agents, and this is we'll spend a little bit of time talking about. So what are virulence factors? Last alternative approach that is used for finding new antimicrobial agents, which is called anti-virulence. So what are virulence factors? Virulence factors are factors that are produced by the pathogen in order to be able to cause infection in the host and cause harm in the host. So this include adhesion, something that the bacteria will use to adhere to the host cell. It includes toxins, which is something that the bacteria will produce in order to cause toxicity in the host cell. Secretion systems is like a system through which the bacteria can secrete its toxic molecules to the host environment. Siderophores, they are molecules that are used to capture essential elements like iron from the host environment. So iron is very limited and very restricted inside the host cell. So the bacteria, if it's able to capture it, this will give it a big advantage. Immune evasion and modulation of the immune system, as we have seen, this is a very important strategy. In order to protect itself from our immune system, it can modulate this immune system. They can help it form a biofilm. They are communication systems that we call them quorum sensing mechanism, and also they could be a very important biosynthetic enzyme that's required for causing infection inside the host. So if we can target this virulence factor, we will disarm the pathogen. We will cause this pathogen to lose its weapons that's causing infection inside our body. So how we can do this? For example, if we have a pathogen that's causing infection because it is producing a toxin, so we can interfere with the transcription of the toxin on the level of RNA. Or we can allow the pathogen to form its toxins. This green cones is the toxin. If this toxin bound to its receptor in the host cell, it will cause toxicity. Now, if we produce a drug that can capture the toxin while still outside our cells, this will protect our cells from the toxic effect of this toxin produced by the bacteria. Adhesions is very important for the host colonization. For the pathogen to cause infection, it needs to be able to adhere to our body. It uses adhesion. So here is the bacteria, and here is the adhesion, and it's binding in our receptor. If we can block this binding, this will prevent adhesion. Another approach is to mess up with the adhesion senses itself. For example, bacteria use this long structure, which is called pili, to adhere to our cell. If we develop a drug that mimic this subunits, so the subunits of the pili is the blue circles, and our drug is the red circles. So if our drug was looking like the pili subunit, what will happen for the bacteria? While it is synthesizing its pili, it will incorporate some of the molecules of our drug. So it will produce a defective pili and will not be able to adhere to our cells. Also, a very important system inside the host, inside the bacteria, is what we call it signaling system or regulatory networks. The bacteria have regulatory proteins that center certain signals and regulate a lot of processes. Can you create virulence factor, colonization of the host, biofilm formation? If we can interfere or block these signaling proteins, we can interfere with all the downstream processes, preventing them from causing harm to our cells. As you can see, anti-virulence drug is a very promising one. It has multiple approach. So let's have a look about the pipeline. We said the pipeline is a place where we have new drugs that are potentially coming to the market soon. However, the image or the situation is not very looking good. Here, in order to have only one molecule FDA approved as a medicine that you can find in the drugstore or in the hospital, in the bedside of a patient, you need to start with all these compounds. You need to pass with all these phases. You need to test on tens, hundreds, and thousands of volunteers. You need to spend all this time, and guess what? Spend many dollars or pounds or whatever. It is a very huge investment. And as we said earlier, that pharmaceutical industry is not interested to spend all of this to just to get one drug that in few years it will be done working in the clinic. But let's see what is in the pipeline now. This is a survey of the compounds that are potentially could be in the market in the near future. If we classify them according how they work, the majority of them are the traditional one, targeting essential targets inside the bacteria. So this will be normal antibiotics, potentiators, repurposed drug. The most proportion or the highest proportion is for antibodies and vaccine. Immunomodulators is small component. Anti-virulence gaining ground, and also phages and microbiota modulation is gaining ground. So there is hope there, but again, as we see here, like we need thousands and thousands of molecules to start with in order to get only one that will work. So it is a huge investment, and we need to be very patient. Good news, we have already FDA approved drugs in the market that are based on virulence and immune body. Here we have the Clostridium botulinum, have FDA approved anti-virulence drug, which are basically antibodies against the botulinum toxin. The same case for the Clostridium difficile, also anti-virulence drug. We have a monoclonal antibody that's directed against the toxin of the Clostridium difficile. So those are already drugs in the market. So now we'll come to the last part. Can we do something like this in our labs? Yes, I will tell you briefly about a story of what we call a homemade drug. We call it Staquorsin. Staquorsin stands for Staphylococcus aureus Quorum Sensing Inhibitor, and we'll talk about it in the next few slides. So the first question we want to ask, where we should look for potential drug target. An ideal position for this is what is known as the host pathogen interface, the place where the host is trying to fight infection, and at the same time the pathogen is trying to establish infection. Here the host is putting its heavy weapons, and also the microorganism is putting its heavy weapons. So one of the very significant battlefield is here, what we have here, which is called the abscess. Here we have a kidney of a mice, and this is an abscess which is caused by Staphylococcus aureus infection. So this is where to look. How about how to look? How we can look at this position? Here we use two technologies. One of them is called magnetic resonance imaging, MRI. I think maybe many of you know about it. Usually you get inside this machine that's like a tunnel, and you get this imaging. Here we are recording the development of the abscess formation throughout 4 days of infection, and as you can see, we can see it. Also, we are using what we call it imaging mass spec. We are getting the very
fine details of the infection as it happens, and we are analyzing it by mass spectrometry to know which molecules is which. So we ended up by having this three-dimensional model. That is the kidney of the mouse, and we can see the place where we have the abscess form. Now we want to get the fine details, so we used microdissection in order to capture this tissue and analyzing by proteomics and mass spectrometry to know what are the host proteins and what are the bacterial proteins that are found in this tiny battlefield, which is the abscess formed inside the kidney of the mouse. So the major identified target in this was AGR system. So what is the AGR system? The AGR system is what we call it a quorum sensing system. It consists of several components. We have a receptor on the surface of the bacterial cell, and it binds to, here we have the receptor on the bacterial surface. We have a regulatory protein that encode system regulatory molecules, RNA3, which activate virulence factor, hemolysins, proteases, lipases, and at the same time it prevents what we call the adhesion or surface associated proteins like fibronectin and protein A. So most of the Staphylococcus aureus proteins found on this abscess was linked to this AGR system. So could it be a potential target? Let's see. So here we have what is called a wild type, a bacteria that has working AGR system. Genetically, we knocked out the AGR system. We have a mutant in the AGR system, and we infected mice with the wild type, and we infected mice with the mutant. As you can see, the wild type bacteria is causing a major infection and ulceration in the back of the mice. However, the mutant lost its virulence capability, and the mice survived this infection. So this is a genetic intervention. We got inside this bacteria and deleted the AGR gene. We cannot do this in the clinic. We cannot extract every Staphylococcus aureus infecting human and delete the gene there. So can we do this chemically? Can we find a chemical compound that can block the AGR system, so we can end up with the same situation using a medication that you can administer either orally or intravenously or topically? So let us see. So we developed a molecule called Staquorsin, and we call it Staquorsin. And as you can see here, this is an in silico prediction for the Staquorsin molecule, showing that it docks, it binds very well inside the active site of the AGR protein, which is the regulator of the AGR system. This is something good. This is in silico. How about in real life situation? Is this compound toxic to the bacteria? Will it gross defect? Will it impose a selective pressure on the bacteria to develop resistance because it is targeting an essential molecule? Guess what? We grow it up to 600 micromolar. Usually we are treating infection in the nanomolar or in the very small micromolar range. 600 micromolar of Staquorsin, either on a methicillin sensitive strain or a methicillin resistant strain, they are growing normally as if there is no Staquorsin at all. So this was very promising, because this tells us that if it will be successful in treating infection, it will not cause this selective pressure we talked about it earlier and cause the development of resistance. Then we tested, is it like affecting the virulence capability of the Staph aureus in vitro? As we said, the AGR control lipases. Yes, it prevents lipases. The AGR control lipases. Yes, it is preventing lipases. And also the RNA3 molecule, which is the effector molecules of the AGR system, is downregulated by Staquorsin. How about biofilm formation? As we said, like, let's go back to this slide. As you can see, AGR activate hemolysins, proteases, lipases. Staquorsin inhibited all of this. AGR blocks binding or adhesion, so biofilm formation will not be affected by an AGR molecule. And as you can see here, again, using Staquorsin, we did not affect the biofilm caused by Staphylococcus aureus, indicating that we are directly acting on the AGR system, blocking its function. How about repeated exposure? This is the problem we have, that all the time when we are using the antibiotics multiple of times, we get resistant. Here we pass the microorganism in the presence of Staquorsin, in a very high concentration of Staquorsin, for 20 times. And as you can see, still we are having the same inhibitory effect. Here is like the passing away from drug and passing in the presence of the drug, and always we get inhibition of the RNA3 molecules. Taking it to the in vivo level, mice were infected with Staph aureus. When they started to be treated with Staquorsin, they started to gain weight and perform better. At the end of the infection is totally cured. If we took this lesion and count the number of bacteria present inside this lesion, we find that we have a significant reduction in the number of the bacteria inside the lesion, indicating that this lesion is free of microorganisms. How about the cytotoxicity of Staquorsin? Is Staquorsin toxic to the drug? As you can see, we used up to 100 micromolar of Staquorsin, and we still, the percent viability of human cell is 95%. Actually, there's maybe no change from this point. So this is very, very promising molecules that hopefully will be able to develop into a useful drug. So let's conclude our talk today. So the take-home message: antimicrobial agents, especially antibiotics, is the focus of our talk today, have saved and continue to save lives all over the world. The need for new antimicrobials is urgent and very critical, yet the discovery of new one is facing technical and financial hurdles. Alternative antimicrobials discovered through biomolecular approaches like we talked about represent a hope, but it still need to prove their efficacy. Cooperative work between academia, industry, and healthcare providers is needed to overcome this crisis. Finally, I will end by thanking the people who contributed to this work. The work was done by Noran, which is the graduate students working on the Staquorsin, Dr. Riham, who is the chemist who developed the Staquorsin molecule, who collaborated with Dr. Eric Carr in Vanderbilt, and the funding of this was from the Faculty of Pharmacy, Cairo University, and also from the STDF. And I am ready for any questions if you have for this talk. Thank you very much.
Thank you, Professor Atia, for the valuable presentation today. So now we would like to have a discussion session, and we would like to have some questions from the audience. Maybe, yeah, for the audience, if you want to ask a question, you can raise your hands first, and or write in the chat. Anyone? Okay, so yeah, okay, yeah, student Ahmad Shahrul, yeah, yeah, okay.
Yes, ma'am.
Yeah, okay. Yeah.
Excuse me, Prof. I want to ask something about biological weapons. In modern warfare or asymmetric warfare, weapons of mass destruction or biological weapons are used.
Excuse me,
I'm not getting your question, so can you like clear it a little bit?
Oh, okay, sir. Excuse me, sir. In modern warfare or asymmetric warfare, weapons of mass destruction or biological weapons are used to attack a country. What if a country makes biological weapon from superbug to attack the other country? What should we anticipate in using these weapons? Thank you, sir.
Okay, so maybe it's like a little bit away from the topic of our talk today, but yeah, if we are not protected, if you are the country who is being attacked by a biological weapon, if you don't have enough medications, effective medication against things, so you will lose this thing, because they are, as you said, they are weapons of mass destruction, so they are will kill a lot of people in a very short period of time if they are not well prepared. So if people are well vaccinated and also have available functional healthcare system that can provide medication that can treat this infection in a very quick and short period of time, keeping in mind that many people will be infected at the same time. So yeah, they will survive. Otherwise, I don't think they will make it. Does this answer your question?
Yes, sir. Thank you, sir.
Thank you.
Yeah.
Next, anyone? Yeah, this is a question, yeah, from the chat. Okay. Many new antibiotics are still developed from the old classes of small molecule antibiotic. And we make a better spectrum and a better kinetic profiles. Is it helpful in terms of avoiding resistance?
Okay, it's helpful for finding having new ones, but I think the problem we are having with traditional one is that they are destined to have antibiotic resistance with their long-term use. So it's good to have, and as you can see when we check this survey of the pipeline, almost half of them are small molecules. So we are still trying to find small molecules that will kill or inhibit the growth of the microorganisms. So I think all those people are not wasting their time and effort to find something that's not useful. However, I think it will be a short-term solution. It will face the same problem, and you might end up like by getting to the industry and trying to convince them to produce this compound for you, and they will refuse because they will not make money out of it. So it's important, yeah. However, it's short term, and maybe it will be the only solution we have if those alternative approaches did not prove that they are like able to replace the antibiotics. But here we are in the post-antibiotic era, so we need to keep looking for them.
Okay, thank you, Prof. Atia. And yeah, I'd like to have one question, and about the phage therapy. Yeah, is that the principle of the phage therapy is to infect the microbes that infecting our body with the phage, or maybe genetically what to say, modified phage. Yeah.
So actually, phage therapy is a very good technique or good approach. It has a very good advantage, and at the same time it is a big disadvantage, which is what we call high specificity. Like if we have a patient who is suffering from certain infection with microorganism, if we isolate this microorganism from him and try to find a very specific phage that will kill this microorganism, and we found it, this will be a cure. And actually this happened already, and many papers are published on this basis. Like they are very highly specific. However, if I got infected with another bacterial strain of the same species, this phage will not be effective. So the high specificity or high strain specificity is good to treat case by case, and maybe this is the future, is what we call it, like precision medicine, like or home tailored medicine. Like we have a drug for the treatment of each case separately. However, it will not be able to use for massive treatment. You cannot have a phage that will fit all the strains of the bacteria. And to complete the answer of your question, yes, this virus or this phage will infect this bacteria, cause infection in my body, and kill it and get rid of it. And again, if it's very specific, it will get rid of it very efficiently, and this patient will get cured. However, we are not sure if we get infected with another strain of the same species 2 weeks later, it will be effective. Maybe no, because it's highly specific. So usually what they do is use a cocktail of phages, like give a collection of phages that could be specific to multiple strains of the same species. I think this is the approach they are using now.
Yeah, thank you, Prof. Atia. Yeah, this is a new insight for us. And yeah, and one more question, Prof. Atia. So about the AGR quorum sensing,
Yes.
is this system is specific in Staphylococcus aureus, or this system is owned by other species or microbes?
Yes, it's more common in what we call them gram-positive bacteria. Excuse me, but they are specific, yeah. Specific to Staph aureus. Like I didn't present this data, but there are inhibitors for the AGR system for Staph aureus, and for example will not affect the AGR system of Staph epidermidis, which is one of the normal microbiota present on our skin. So this is another selectivity in order to just target the virulence microorganism and not affecting the normal microbiota. But the system is present in multiple species, yes.
Okay. Thank you, Prof. Atia. Maybe another question. Yeah, from student or faculty member. Okay. For student, yeah, Indonesian question would be okay. We will try our best to translate it. Yeah, if you have a question, yeah, you can ask in Indonesian. Yeah. Okay, so I think the time has limited our meeting today. So yeah, we would like to thank to Prof. Atia for kindly give a lecture today. Yeah, this so many new insight about antibiotic or antimicrobial agent. Yeah, and yeah,
It's my pleasure.
Yeah, thank you for the lecture today, and for the audience. See you on the upcoming guest lecture with Prof. Atia, and in after Ramadan. Yeah, so thank you. Assalamualaikum warahmatullahi wabarakatuh.